I've got a bachelor and master's in kinesiology from University of Waterloo. I've did a PhD in neuroscience from University of Montreal, and I did a postdoc in University of Alberta. All of the last two, I worked with level of spinal cord, in the sense that at the PhD level, I looked at spinalization of a cat and retrained it to walk. In Alberta, we did functional electrical stimulation for helping walking, and also for bone density, which is a big problem which you'll have to deal with something going to space. I'm at UQAM now, Université Québec à Montréal, and my projects are looking at various neural function in locomotion, in sexual function, and also with older people. So today, what I'm going to do is, since you're dealing with a population or a group that either goes into space or earthbound, but with some pathology or some problem with the nervous system, or what I decided to do is that, why the title is, look at how the body itself controls the movement, and what can you do, how can you use these information to help you control an exoskeleton. So what I'll do initially is, here's the sort of outline of the presentation. So I'll give you a brief example of control, which you're probably familiar with, but it's very little. And then I'll talk about the natural sensors, mostly internal sensors, because I mean we have, as you see, we'll see, we have external sensors, or sensors that do capture information from the outside. And then I'll talk about the motor response, basically reflexes when you trigger these sensors. And then I'm going to come back to how can you use this information to control an exoskeleton, and what do you have to look at in terms of this information, because you're dealing with a body that's live and will have these responses. So it's not like you're building a robot. You're building something, essentially an orthosis that attaches to a being that has its own control. So you have to be able to take care of that. So essentially, if you look at control of movement, you have essentially a central command. So in other words, a motor plan and algorithm that generates the basic pattern of movement. And then you need to adjust these to various situations, whether it's uneven terrains or different perturbations that occur. And you can also use this information, these adjustments, to go back and retrain the motor program, or correct the motor program that you've outlined. And I just took this picture from the presentation that Adrienne had given you the other day. And in this particular situation, you're using EMG signals to drive some model, and you're using joint angles and to control the model. You'll see the sensors that I'll present to you today will give you some of this information and more. And you can use them to control or to assist your system. So if we take, for example, a simple, very simple task, such as opening a door from a standing position. Well, if you look at what needs to be done is, first of all, you need some control of the postural muscles. So essentially, you see from the little diagram, the activity of the muscle, very schematized, is it's turned on in the leg muscle and the paraspinal muscle way before the arm muscle. So in other words, you stabilize your body so you can actually do the actual movement that you require to do. Of course, there's also fine-tuning that's added on top of with the prime mover. And if you combine that with in dynamic situation, which is not a very like this particular example is very static, but you need to consider the dynamic situation. Now, and in the dynamic situation, obviously, we'll have to have on-the-fly adjustment. In other words, ongoing feedback to either help control and add. And in order to do this, you need to know where the body parts of the body is in relation to either the space that you're working in or the segments with respect to each other. So in terms of control, so we have some central command coming from the central nervous system, for example, that is sent to the muscle to control the muscle to activate the various parts. At the same time, you have a copy of this information that is sent to the central nervous system. In this case, it would be the cerebellum. I've just put these boxes very general for now. And then you have all these sensors that telling you where the body parts are, or to help you control the movement. This information is sent back to the central nervous system, and that gives you essentially what is called proprioception, to control movement without the use of visual system. Because of course, when you control an exoskeleton, for example, you could have cameras, but sometimes it's not the most appropriate, and the treatment of the signal requires a lot of information, whereas the other sensors can be a lot easier to work with. So if you look at the sensors that are used for orientation of the body in space and segments with respect to each other, and the movements, you have sensors that are get information from the exterior. For example, as I mentioned, the visual system, and as I said, it's not always available. You have also the auditory system. Both of these I won't cover today. I'll look more at the proprioception. In other words, the sensors in the muscle, in the tendons, the cutaneous sensors, the joint receptors, and the vestibular receptors. All of these provide a certain amount of information, and you'll see there can be redundancy in the information. First of all, what is proprioception? Proprioception is the perception of the body or the movement, and also force of the limbs or body segment with respect to each other, the WRTs with respect to. And it gives you information also about the orientation and movement of the body in space. Obviously, it's necessary to maintain equilibrium, but also controlling the movements. And in addition, if you want to manipulate objects, it's also very important to have the information, the proprioception information, to be able to use that information in 3D, to evaluate forms of objects in 3D. There's two submodalities, and I won't separate them too much. One is stathesthesia, which is the position sense. In other words, it's static information. It tells you the position of the limb with respect to each other, not during movement, whereas kinesthesia, the kinesis meaning movement, it gives you dynamic information, both speed and direction. In some cases, it'll give you acceleration as well. So you can use some of that information to control your exoskeleton. Again, what we'll first look at is the muscle. We'll look at each one of these receptors and what type of information, in other words, the boxes in blue, what type of information they provide, and eventually what are the motor response when you trigger or you activate these sensors. The first one is muscle receptors. There's two muscle receptors. One is the muscle spindles, and they're in parallel with the muscle fibers, the external or the extrafusal muscle fibers. And then you have the Gogli tendon organs, which are at the junction, the musculoskeletal junction, and they're in series with the, like as you can see here in the blow-up, they're in series with the muscle fibers. In other words, a number of muscle fibers attached to these tendon organs within at that junction. So what does the muscle spindle provide? If we blow it up and schematize a little bit this setup, you have some other little parts inside, which are the sensors, and you have nuclear bag and nuclear chain. And the 1A here and the type 2, these are afferent fibers. They conduct information from that sensors towards the central nervous system. The first one, the 1A, being attached in the central region, okay, will transmit information about the rate of change of muscle length. In other words, it gives you dynamic information, as if the muscles are actually moving the limbs. The type 2 afferents, coming from more the polar regions of the receptors, they transmit more static information. In other words, they transmit length information. So in other words, they gives you the position of the joint in that area. The Golgi tendon organs, they have the type 1A afferent, as I mentioned, they're at the musculoskeletal tendinous junction in line with the fibers, and they transmit muscle force information. So in other words, they inform the nervous system about the force. What's important also is that they're more sensitive to a contraction, in other words, the force given during the contraction, than during just simply stretching the muscle. Okay, so they give you more information when it's actual contraction of the muscle. The cutaneous receptors, okay, as you can see, there are a number of them, and they're in different parts of the epidermis and the dermis. Okay, so if we look again, schematized, in the epidermis, there are Merkel discs and free nerve endings. In the dermis, there's Meissner corpuscles, Krauss corpuscle, hair follicle, again free nerve endings. In the dermis, the deeper layer, the Ruffini corpuscles and the Pacinian corpuscle, again with free nerve ending. And each one have most of them are active but type 2 afferent, similar to one part of the muscle spindle, and there are other types of afferents. I'll come back to that, what that means in a few minutes. If we look at the Merkel disc, located in the epidermis, like the purple ones here, they inform the body about light touch and also light pressure. And they're also used for texture discrimination. So in other words, if you have, for example, silk versus sandpaper, then you rub your hand and it'll help you with these sensors. They're sensitive and they'll be able to decode some of this information. They're slowly adapting receptors. In other words, they respond mostly to a duration that's kept on for a period of time, as opposed to one that you'll see in a minute that's more rapidly adapting. These three sensors, the Meissner corpuscles, which will be here, the Krauss corpuscle, which I don't have on the diagram, and hair follicles, they all give the same type of information. This is the type Meissner, Krauss, and the hair follicle in that order from left to right. They inform light touch again, light pressure, and vibration. So in other words, anything that's up to 100 hertz, their most sensitive is around 30-40 hertz. That's where they respond the best. And of course, they tend to give information about the variation of the stimulation. The Ruffini corpuscles, they're in a deeper layer with the Pacinian corpuscle, and they inform touch, but very vague, and pressure. And they're sensitive also to, if I stretch the skin, then it's sensitive to that stretch, and also sensitive to the direction of the stretch. They're also slowly adapting, so like what is SA, slowly adapting type 2, and they give information about, again, the stimulus duration because they're slowly adapting. And then you have Pacinian corpuscles, which look a little bit like an onion with many layers, and they also respond to pressure and vibration. And vibration is quite varied, with optimal, from 30 to 1500, and optimally the best is around 250, 300 hertz. So it responds to that, and it's a stimulus. It responds to stimulus variation. In other words, on-off type of thing. The free nerve endings, wherever you see the free nerve endings, in other words, there's no particular capsule or ending to it. There's simply an axon that has no terminal on it. They indicate pain and temperature. And of course, because they can also indicate a very vague touch without necessarily given precise information. So if this is sort of a summary slide of the main sensors that I've showed, except for the free nerve endings. So in other words, they respond if you have in this case, you have the localization of the either superficial or deep. You also have on the bottom the adaptation speed, rapid versus slow, and then also receptive fields, how wide they cover the skin for example. So the ones that are more superficial, they're more, they tend to be very limited, so that you would need more to cover the whole area. Whereas the ones that are deep, they tend to have a large receptive field. And also you see the response, the axon discharge from a simple stretch and then a release. So the ones that are rapidly adapted, in other words, they tend to be off and then on when there's something happening, and then off when it's maintained. Whereas the slowly adapting, the ones that are here on the right, they respond a little bit during the changes of stretch, but tend to maintain also activity during the static position. So these tend to indicate sort of some sort of position or object being placed, for example, in the hand and maintained there. Whereas these, the ones on the left, would respond to when the object will be placed and then removed. So only the on-off area, whereas this one is continually on. Joint receptors, there's 4 types, and they're very similar to the cutaneous receptors in many ways. They're given numbers as opposed to a name, but you'll see that they're also very similar. They have the name similar to the cutaneous receptors. And this I'll go into what that means in a few seconds. So in other words, if the first one, the type 1 receptors that are found mainly in the capsule of the joint, they looked a bit like Ruffini endings, and they indicate the speed and the direction of movement, particularly at the extreme. So they're there to indicate when you're reaching the end of the range and beyond which you're unsafe. In other words, you could damage the structure. So they indicate this at the ending of the movement. They also provide constant information. If you maintain it at those points, then it still maintains that you're in a dangerous zone, for example. The type 2 receptors, or Pacini-like, they look like Pacini. They have rings like onions. They respond to small movement and acceleration of movement. And they're found both in the capsule and in the ligaments. And their response is each receptors will have a particular angle which it's optimized for. But it doesn't tell you which way the movement is going. For example, if you're doing a flexion of the elbow or an extension of the elbow, these receptors will respond, say, for example, if we take the first one here at the left, it's somewhere around 75. It'll respond optimally around 75. And after that, around 90, it will shut off. It won't provide any more information. So these provide specific angles, but very few details about which way the angle is going. The type 3, they look like Gogli tendon organs. They're Gogli organs. They also indicate the direction of movement and the force applied, and they're found in the ligaments, so the force applied to the ligaments. And a bit like the type 1, they're more at the ends. So they respond when there's actual stretching of the ligaments. So they're there to protect also the ligaments. We saw that the type 1 tended to be in the capsule, and they responded to protect the capsule from being stretched beyond its safe limits. Whereas these type 3, they're more in the ligaments. Finally, for the joint receptors, you have again the free nerve endings, and they're sensitive to pain, and they're found all over. They're found in the capsule, they're found in the ligaments, they're found near the bones and in the bones. So whenever there's damage to the structure, they will send information back to the system. When I talked before, I talked about the type of afferent fibers, the information that's being sent back. Each one has a different speed of conduction. So in other words, information will reach the nervous system either faster or slower depending on the type. So in this category, at the left, is if you look at the diameter and also the speed of conduction, which is in meters per second, you can see that muscles, either the muscle spindle with the 1A afferent and the Gogli tendon organ with the 1B afferent, they conduct very fast. The cutaneous receptor as well as the secondary muscle receptor, which have this type of afferent, type 2 afferent, and also the joint receptor also have the type 2 afferent. They conduct a lot slower, so it takes longer for the information to reach the nervous system. Then you have type 3, or in this case, they tend to be either nociceptive, in other words, pain receptors, or thermal receptor, and they conduct a lot slower. And finally, the slowest types of fibers are type 4, and they're, like all the other ones, are myelinated. In other words, there's saltatory conduction, so it's very fast, or faster. Whereas this one is not myelinated, and it provides slower conduction speeds. Here we're talking about 0.5 to 2 meters per second. So it's very slow if you compare it to the 1A or 1B or even the type 2 fibers. And these are, again, pain and also temperature receptors. The difference is one in the type 3 and type 4, this would be the equivalent in pain, would be equivalent of pinprick, whereas the C here would be the equivalent of a burn. They're different speed or different information that's being transmitted. So all this information from these receptors, whether in the muscle, the joints, cutaneous, and temperature and pain, they're all transmitted to the central nervous system through the spinal cord, to the brain stem, and eventually to the cortex to be evaluated and perceived properly as being not a problem or being a problem. And the pain receptors and temperature receptor have a different path, but eventually make it to the same area of the brain. They use the pain and temperature receptor, what is called the spinal thalamic tract, whereas the joint receptors and the joint receptor, cutaneous receptors, use the dorsal column. So in other words, here in the dorsal column, they ascend to the brain stem and eventually get connection all the way to the cortex. The vestibular receptors, they're found in the sort of middle ear, the inner ear, I should say, and they're composed of two parts. They're semicircular canals, and there's the otolith. If I bring it to this one, so in other words, you have three canals that are paired on each side. So you have horizontal, posterior, and anterior, or sometimes called superior. And you have the otolith. The canals transmit acceleration, angular acceleration information. In other words, if you tilt your head, then it detects this acceleration, or if you turn it, rotate your head, it's the same thing. Whereas these tend to be linear acceleration, either vertically or horizontally. Of course, when you combine the two, you have different angles, but essentially they're linear acceleration. That information, compared to what we've just seen before, is sent to the brainstem, in this case the vestibular nuclei, and from there the information either sent down or sent up. I just showed the sending down through the lateral vestibular tract, vestibulospinal tract, or the medial vestibulospinal tract. So they tend to go down towards the limb muscles at various points. So what type of information do they transmit? They transmit, as you saw, acceleration, either for the pitch, roll, or yaw in terms of control, and also linear acceleration. In other words, if you're, for example, in a moving car or something like that, then it gives you this information, or an elevator going up and down. And essentially, it helps to determine the body, or the head in this case, particularly the head, its orientation in space. So it gives you this information, whether it's inclined or rolled or whatever. What this information then can be used, and it triggers motor response. If I activate, for example, the vestibular receptors, or the cutaneous receptors, or the joint receptors, for example, I will trigger a response through the afferent fibers, as I suggested, and that will trigger a response. And these are called reflexes. They're there initially to protect the body. Okay, the response are there to allow the body to respond as quickly as possible without necessarily having to reach the brain and the motor cortex, for example, to wait for a response, because sometimes it's too late. The best example of that is when you have, if you grab a cup that's very hot, your response already taking place before you notice that it's hot. So they have to be there to protect. So there, I just outlined here two you have the spinal reflexes. In other words, some stimulation occurs either from a skin receptor, in other words, cutaneous receptor, muscle receptor, or joint receptor, through its afferents, whether it's type 1A, type 1B, or type 2. It's going to the spinal cord. It makes connections with the motor neurons in the spinal cord and sends that information to control muscle groups. Now that's a spinal reflex. You have the same thing, but this time it can go up to higher levels. In other words, that information keeps being transmitted, as you saw in the diagrams before, to the brainstem or even the cortex. And so that'll give you what's called supraspinal reflexes. So if you look at, in this case, if we look at the one of the type of reflex, you would have a stimulus that's here. You activate this receptor. You get a response, which is the spinal response. Then you get the supraspinal response, and eventually you get a voluntary response to that stimulus. In other words, you actually react to the stimulus. But you've noticed that there's two responses that occur before that for this particular type of sensor. In this case, it would be, for example, the muscle spindles, which are the fastest one, as I mentioned before. So the first reflex, and you've heard of this reflex before. It's the equivalent of the tendon tap. You go to the doctors, he taps you on the tendon, and you get a muscle response, a movement of the limb. So in this case, for example, if I tap the Achilles tendon, then I activate the muscle spindles, which transmit the information to the spinal cord, and then comes back through the motor neurons and controls muscle groups. The response of that tap means that you'll activate muscle, the monomus muscle. The monomus muscle is the muscle from which the receptor originates. In other words, if I stretch the soleus muscle, then the spindle in the soleus will trigger a response, and it will come and activate the soleus muscle. It also excites the synergist muscle. In other words, in this case, the gastrocnemius muscle, which would be synergist to the soleus. But it can also do the quad as well as a synergist muscle. In these cases, they're monosynaptics. That means they have one synapse at the level of the spinal cord. In other words, the afferent makes a direct connection to the motor neurons that controls that muscle. So it's very quick for that. And then there's the inhibition of the antagonist muscle. In other words, you're closing or stopping the antagonist muscle from reacting at the same time, so that the movement can actually occur. And these are through an interneuron, which is called the 1A interneuron. Here on the diagram on the left bottom, what you have is an H reflex, which is the same as the tendon reflex, except that we use electrical stimulation instead. It's triggered, so we bypass the whole spindle aspect. The tendon tap, which is what we've shown, and the stretch reflex is, instead of being a tap, very steady, sudden tap, is you could have a stretching muscle. You push, in this case, you would push on the foot and you would stretch the muscle. So as you can see, these different would have different latencies. So if it's an onset very quickly, for example, if I stimulate with electrical stimulation, it's faster than the tendon tap, than the stretch reflex. What this particular slide shows is, particularly here on the right, is that reflex is not constant. You would think that, and it makes sense, because if you think the reflexes are there to protect, then they should be modifiable or adjusted. So here is the H reflex, which is the same as the myotatic reflex, during walking. So you can see that it's very high or higher. This is the stand space. and this is a swing phase of the movement. So in other words, it's totally shut down during the swing phase. It won't respond as it would during the stance phase. So it's modified according to the phase of the step cycle. And on the left is a diagram showing what can come and influence some of the interneurons and the neurons, either from the descending or information coming from the other muscles. How it could come and change what the results will be. The tendon organs have, what they do is you stretch the tendon, and it'll provide through an interneuron, in this case 1B interneuron, it'll inhibit that particular muscle. Remember, this is force information. That means that if you're stretching, you're providing perhaps too much force, and so what it'll do is it'll shut down the muscle. It'll shut down the synergist muscle, from preventing them from being activated, and it'll activate the antagonist muscle. So through again interneurons. The cutaneous receptors will provide information that is a little bit longer, and if we go to the next one, you can see that it'll excite. For example, here the stimulus is at the tip of the toes. It'll excite the flexors as to remove the limb away from the offending stimulus, and it'll provide inhibition to extensor to allow the flexion to take place. Whereas on the other side, on the other hand, especially if you're standing or walking, then it provides what is called a cross extension reflex, and you'll have extensor being activated and the flexors being inhibited. So it allows you to stand on the left foot, in this case, when the right foot is being removed. As with the muscle reflexes, they can also be modified. Again, this is from the work of Yang and Stein in the 90s. What you see is, again, phases of the step cycle, and you can see very little response at phase 6, which is somewhere around the transition of stance to swing. During swing phase, there's a big response in the middle of the swing phase, and a bit further on, just before, that would be just before contact again, it's inhibited, the response. So again, as with the muscular reflex, it's adapted to the phase of step cycle. Okay, it could be adapted to other types of movement. In this case, it was walking. It's been shown both for muscle receptors and cutaneous receptors, bicycle movement, whether it's arms or legs, you get this modulation as well. The vestibular receptors via the lateral vestibular spinal tract will provide a response that is, if the head is brought forward, then you'll get an extension of the upper limbs and flexion of the lower limbs, as if you're falling forward. When on the other hand, when it's backwards, you will have flexion of the upper limbs and extension of the lower limbs, as if you would go and grab a bar or something to prevent you from falling backwards. There's also the symmetric tonic neck reflex, and this one is activated through muscle receptors that are in the neck, and they're sort of opposite to what we just saw. In other words, before we had head movement, but this one is neck movement. So a flexion of the neck will provide flexion of, which was down here, flexion of the neck, will provide upper flexion of the upper limb, extension of the lower limbs, and extension of the neck will provide extension of the lower limb, which is at the top here, and flexion of the lower limb. Okay. What's interesting is that these two can be combined. This is the tonic vestibular reflex, and this is the symmetric tonic neck reflex in this way. On this quadrant, what you have is the addition of the two with extension of the head, of the neck, and flexion of the head. Or in this case, it's the same thing, but you have the opposite effect. On the other quadrants, you get negative effects. In other words, they subtract each other or they cancel each other. So you would have a flexion of the neck and head forward, you would have cancellation, which would look more like the neutral for everything. So they can be added together almost algebraically. Then you have an asymmetric tonic neck reflex, again the muscle receptors in the neck, and in this particular case it's with respect to the rotation of the head, again provided with also the neck and vestibular receptors. What you have is extension of the limbs, upper and lower, in on the facial side. In other words, where the face is facing, and then on the occipital side, on the back of the head, you have flexion. And this you could see this effect very well in cyclists when they're, for example, if you Tour de France or Olympics, when these guys are really tired, they start to oscillate the head back and forth, and they're actually using these reflexes to enhance their limb movements. So if we look at the control of exoskeleton, you could use the feed forwards. In other words, you send just a command, a motor plan that dictates the instruction of what muscle, what things should be done. And you use feedback for immediate correction and also to modify that motor plan if there's errors in it that needs to be corrected for the next time it'll go ahead. So now, how can we use the information from the sensors to either enhance or negate some of the effect? First of all, you could use the natural sensors either by recording their activities or using implants, again, to record, but on the nerves themselves, they conduct information. Again, it could be used as feedback or feed forward to adjust the movement. It can be used to enhance the movement. In other words, if I need a little extra boost, so instead of having the exoskeleton just do all the work, I could use the remaining parts of the body to enhance the movement by triggering, for example, a reflex response. So that will enhance the movement. So, and with the effect of that will be that you will require perhaps less external power to control the skeleton because you're using the remnant parts of the body to activate and to move, as opposed to being complete robot-like activity. But you also need to negate or minimize the negative effects of these motor response. So one is from one time to the other, there's variability in the response, and there's plasticity, as you saw, and in other words, modification in the control of the movement. And it could be habituation to the type of, if I repeat the stimulation over a period of time, I could actually have habituation to that, and then the response is decreased. That could be a good thing in a way that once it's habituated, you tend to get more of the typical similar response, as opposed to when it's actually habituating during the process, then you'll have change of response. If you're going to use do a normal control, you got to be able to eliminate some of these responses. For example, if you're trying to move the exoskeleton very quickly, you could actually trigger a stretch reflex. So, or a Gogli tendon reflex, inverse myotatic reflex. So that means that the movement would have to be slower as opposed to faster. And if you're looking at the exoskeleton to provide assistance for, say, a pathological situation or injury situation, like a spinal cord or cardiovascular accidents, what you have is these reflexes are enhanced. They're greater than what you would find in normal situation. So a very, very small stimulus will create a big response. So you have to be able to negate that response so that you don't have your system fighting your control system on the skeleton. The other thing with the natural system is the natural body, there's redundancy in the system. For example, if I lean forward or backwards, I could have the stretch of the posterior muscle if I lean forward, which indicates that I'm leaning forward about the ankle, but or backwards if I will stretch the anterior part of the leg. But the same information could be provided by the sensors on the bottom of the feet. So if the front is loaded, then it also indicates that your body has shifted towards forward, whereas if you shift backwards, then the pressure on the hindfoot. You could also have with cutaneous receptors information from the skin being stretched. Joint receptors, again, will also provide either at the end when it's dangerous, and same thing with vestibular, they'll provide information as well as the lean will be provided. What's also important in control is multi-sensor integration, in the sense that if we go back to this one, we see that if I'm only taking information about the ankle, then I could be wrong about the foot or the ankle. Then if I only take information about with the head, in other words, the vestibular system, I could also be wrong because there's a change happening here. So you need information not from one area, but a number of areas. So you have to integrate sensors at various areas to be able to account for these changes. They're two very different situations, but the information here about the ankle and the foot would be the same. And even at the head could be, well, in this case it would be different. So you would have leaning forward, but extension here, and the weight would be more central in this case. And as I suggested, there needs to be adaptability of the system. As I mentioned, there's a number of things that can adapt in this case it's a spinal cord, but in your case perhaps some sort of motor program with an algorithm. So in other words, where the stimulus is coming from, or the intensity of the stimulus, and the type of stimulus that could be changed, and also the joints, whether it's the afferent, whether it's come from cutaneous, muscle, articular, if you're using a natural system. The phase of the movement is important. This all fed back at the level of the spinal cord, and they'll generate some sort of reflex, some sort of movement, which can be also affected by basic neural activity. In other words, I'm already contracting a little. Fear is a problem. If somebody's scared, then these response will be enhanced. The amount of attention you pay for a stimulus, and also the experience and habituation, they also have effect. In other words, if I've had a stimulation for a reflex, the next time won't be as bad because I know what it's like. So that will also affect how you control the system. That's the end of my presentation. Thank you very much for your attention, and good luck with your project.